Hello, everyone, and welcome to today's Drug Hunter Flash Talk. I'm your host, Dennis Koester, and we're happy you're here today. And we will be talking about dose number as a tool in lead optimization in the discovery of orally bioavailable compounds with Abdellatif and Pete from Merck today. So welcome, Pete and Abdellatif. It's great to have you. It's a pleasure.
Thank you for having us.
Thank you.
Yeah. Without further ado, I want to introduce both of our guests, Abdellatif and Peter. So Abdellatif ElMarrouni is currently a principal scientist in the discovery chemistry at Merck in West Point, Pennsylvania. And he's working on drug discovery programs in infectious diseases and neurodegenerative disorders. And he's also co-leading the efforts on implementing medicinal chemistry design strategies using in silico methods at West Point. He received his PhD in organic chemistry from the University of Girona in Spain. And during his postdoc with Casey Nicolau at Rice and Scripps, he focused on the total synthesis of complex bioactive natural products. He's also a member of the Medi division of the ACS. Welcome, Abdellatif.
Thank you.
Yeah. And we also have Peter Wuelfing with us today. He's an executive director in the Pharmaceutical Science Organization at Merck in West Point, Pennsylvania. And he leads the team that's responsible for compound developability profiling, preclinical and toxicology formulations development, and initial first-in-human formulation strategies for small molecules and biologics. Pete has more than 20 years of experience in manufacturing, analytical, pre-formulation, and drug delivery roles. And he has authored and co-authored more than 50 manuscripts and more than 50 conference podiums and poster presentations. He is also a member of the American Association of Pharmaceutical Scientists and an ACS member, and is a board member of the Internal Consortium for Innovative and Quality in Pharmaceutical Development. Yeah, it's a pleasure having you today, Pete.
Thank you.
Yeah. Absolutely. And before we get started, we kind of like to warm up our guests and our speakers with a few icebreaker questions. So I'll ask both of you the questions, and you can decide who wants to go first. So, what inspired you both to start a career in drug discovery?
Chief, why don't you go first?
Yeah, no, I think it's a good question. So actually, I wanted to become a physician, a doctor. I went to study medicine, and they sent me to do pharmacy, and pharmacy includes a year of chemistry, at least in Spain, how the way the studies are done. And when I started doing chemistry, organic chemistry, and all the other, chemistries associated to it, after a couple of years, instead of taking the medicine kind of route, I decided to stay and stay with the medicinal chemistry in that case. And after that, I was very fascinated with, bioactivity, compounds. And it's mostly because I realized that we provide tools to physicians. We're able to invent those tools that can help the physicians to then help patients to feel better, against those diseases. So I'm here now at Merck trying to deliver drugs with my colleague Pete and being able to invent those novel therapeutics.
For myself, I perhaps am unlike many online who are likely medicinal chemists. My chemistry, I studied more physical chemistry at University of North Carolina at Chapel Hill for my doctorate degree, and we looked at electron transfer and gold nanoparticles. So that was not something that naturally was leading me down the road to medicinal chemistry or to a discovery biology role. And so coming out of graduate school, I joined Merck in their manufacturing division. As you can imagine, there's processes, there's chemicals, things go wrong. And I was more of a generalist with respect to chemistry than I was with respect to synthetic work or understanding binding and so forth. So I joined Merck in that capacity, and then it moved earlier and earlier into different parts of the research labs, development labs around formulations, methods, and so forth. And then eventually got to the point where I started interacting with people like Abdellatif and the discovery interface. And that's where my career with respect to developability attributes with molecules came about. So that's how I ended up here. Yeah. And it's great to have an opportunity to contribute to something, to technology that could help people out.
Yeah, no, those are great motivations. I had probably a similar motivation to Abdellatif. Like we're kind of, as medicinal chemists and formulation scientists, we're kind of the first to touch a medicine before it goes out to patients, right? So that's really, really fascinating for me. That being said, what's your favorite molecule and why?
Actually, I love caffeine because I need it, but I also enjoy drinking coffee. So
Yeah.
I have to say that, as being anybody like in this forum, probably we love to see the molecules that we're drinking or intaking and understanding them. And it's just, it's a small molecule. It smells good. Probably it has other components there, but it also feels good, at least from my perspective. So I always like fascinated like such a natural product being able to give us. And how many drinks are on the wall? How many people drink it? And I'm sure there is a long list of benefits. And of course, there is probably risks, health associated with it, depending on how much you drink of it. But yeah, caffeine, go for it.
Good
I'm
one.
going to I'll double down on that. I love it. You're a person after my own heart. Yep, it's a great molecule. So we'll leave it at that.
Sounds good. Cool. So what are you most excited about in drug discovery today?
Well, I think yesterday we got the Nobel Prize. I think the Nobel Prize was probably not a big surprise for some on the field, right? I think we're seeing an impact of technology. We're seeing an impact of how computers are joining, I would say, a science that was more driven in the lab. And
Yeah.
scientists like myself who were training mostly on the lab, but now we're seeing how computers can help us. They already help us every day, right, to get better. Our phones, everybody has a phone, tell you where to go, what's the weather. So why not, use those tools to help us, to guide us in what's the next molecule, what's the next target, how can we do better, and especially going faster. So I'm very excited on seeing how the technology, comes on board and how we come also, to use it as medicinal chemists. And I think it's very exciting times right now for drug discovery in that direction.
Yeah, I'll only echo, right? We've sort of merged fields before, like you mentioned, with automation and wet chemistry and different, merging of different fields. So it'll be really fascinating to see where the computational power takes this field moving forward. So yet another merging of different technologies, and we'll see what innovation it sparks.
Absolutely. Yeah, no, that's a very, very exciting field. Yeah, I'm not surprised to see a Nobel Prize in that, given, all the advances in AI these days. Yeah, cool. So now that, you both have definitely some experience in your career, and I wonder if you could go back to when you started your career. We earlier talked about why you started your career in drug discovery. So if you could go back in time and give yourself some advice when you were starting, what would you tell your younger self?
I mean, I would say listen, because sometimes you have an opportunity in front of you, or you have some kind of a challenge. And my younger myself was probably quick deciding in a no. And I think I would tell myself listen, because you don't know really what that might provide you, right? So it could be in any stage of my career or any stage of my personal life. Is like you listen, you evaluate, and then you make a decision. So my advice to everyone is like, when somebody approach you with something, just take the time to listen, understand it, and then make the decision.
Yeah, and that's great, whether it be a technical discussion or whether it be a career opportunity. Like you said, quite often we're able to walk ourselves out of what could be a really unique way to diversify your skill sets and your experiences. So maybe not saying no so quickly is a good idea, as well as trying to understand yourself is certainly it's a difficult thing to do, and know what you like and what you don't like. So that seems to be, there's so many different people that we all know. There's certain people who want to do one thing and one thing only, and those life in many ways is very okay. They know what they want to do. Other people could be happy doing many things. So you try to find yourself on that continuum and understand what it is what will make you happy in the long run. And so then you need to certainly try to pivot yourself in those directions. But knowing yourself is not the easiest thing to do, and your preferences can change over time. But I would tell myself to keep that in mind, and keep that in mind.
Yeah, no, those are certainly really good advice. I love personality tests too, Pete. So I'm like somewhat obsessed with that testing myself. had changed over time. So and listen is also, I think that was also what I said actually, Abdellatif. So really, I think this is great advice. Last question for you guys before we get started. What do you most like about Drug Hunter?
I mean, I like the email you sent yesterday, which, gives you a highlight of molecules. I can see the structure in your email. I can see a little bit of data, enough for me to get a taste of what I can get before clicking into the details. But those emails, like everybody else, time is precious. So I think having the structure as a chemist, it helps. Having some key information, it does help. So I really appreciate those spotlight kind of emails that we get from you guys.
Awesome.
Yeah, a nimble organization that can highlight interesting topics from across authors, publications, what have you, and bringing it out for the broader field. That's a wonderful service to provide. So that's exciting.
Awesome. Yeah, thank you guys. And yeah, before we get started, I wanted to show you quickly one of the new features that we just recently released by kind of quickly sharing my screen. So what we recently did is we made the INN list searchable. So oftentimes in that list, drugs structures are actually getting disclosed for the first time. So before there's an official first disclosure, those things find their way into the INN list. And we make this available for you so you can kind of search this by a specific target. So for example, if you're interested, let's say, in, let's say, CDKs, you can find out what are some of the recently named CDK inhibitors, and you can find out their structures here as well. So, that gives you an opportunity really to find out a structure before, it's maybe officially getting disclosed. So I think this is a huge list of like really inspiring structures. And one of the things we learned from this list is there's actually a lot of BCPs in the clinic right now. So we're actually going to write a review on that shortly. And with that being said, I now hand it over to Pete and Abdellatif to walk us through their presentation and sharing a little bit more about dose number as a guide in your lead optimization and how you could use the dose number to optimize oral bioavailability in your discovery campaign. It's a pleasure having you again, and we look forward to your presentation, Abdellatif and Pete.
Okay, excellent.
Great. So thank you first, Denise, to get us for the invitation, for the introduction. I think this is a great opportunity to interact with all of you, but also, for myself and my colleague Pete to kind of share where we are in this field and how we have been leveraging it. And I think at the end, we do have the reference. So this is, as you mentioned, a flash talk on this concept. We invite everyone to review that publication and, of course, reach out to us if there is any questions after the flash talk. So the main question is why those numbers and why is relevant to mostly this audience here, which are mostly medicinal chemists, and why is relevant to even myself or to my daily job. So actually, as Pete mentioned, most of us, we deliver a clinical compound or preclinical compound and then takes the way to development. And in development, there are so many issues that I wasn't aware during part of my career. I don't experience it myself, but one of them is how do we make sure that the compounds are being formulated to be able to be taken by patients or in clinical trials. And often one key component that plays a role in those formulations was the solubility of your compound. So if we bring the concept of solubility and we dial down in terms of how can we early on at early stages get a sense of how get some kind of risk assessment on that solubility, those numbers is actually, I would say, a development parameter much more used in those organizations that can be widely leveraged by medicinal chemists very early on by taking some assumptions. And here it comes, right? Now you have a parameter, an MPO parameter, a multi-parameter optimization that we can leverage at very early stages to give us some kind of sense of risk and what might happen to the compound that we are progressing in a development or clinical stage. Pete, you want to take it forward? All yours.
Sure, absolutely. Okay, excellent. So thanks for the kickoff. So there's a couple of good reviews here on the screen. It's by Brown, Wopes, and then Bhutani and Kumar. And you can take a look at the molecules, all the molecules. We'll focus on small molecules today. All the molecules that have been approved from 2010 to 2019, and then a 2015 to 2020 review. And there's a few generalities that you can take away from that. If you look at the plot on the top left, you'll see the green is NMEs, which are by and large small molecules. There's some peptides in there, but by and large small molecules. So quite a few small molecules or small molecule therapeutics are still very much an important part of the mix of new therapies coming forward. You can see on the bottom center pane that by and large those are oral small molecules. So the oral bioavailability question remains certainly important to all of us working in the drug discovery space, looking for small molecule therapeutics. And interestingly, the analysis from Brown and Wopes in the middle, you look at the rule of 5 violations. For the most part, over half molecules are coming forward with 0 rule of 5 violations, which is quite impressive. And then maybe 30% or so have somewhere between 1, 2, or 3 rule of 5 violations. The rule of 5 violations, as you all know, could lead to, amongst other things, a loss in solubility. And so we can, as well as so that speaks to the to the preponderance of some challenging physicochemical matter in the molecules that are coming forward. There was a separate review by Lofton and Brewster that suggested that up to 40% of molecules in the modern pipeline have solubility issues. It would be low solubility by the regulatory biopharmaceutical classification system. If we have time, we can touch on that later. So it feels like, yes, certainly physicochemical properties, solubility still, those challenges are still alive and well. And as Teef mentioned, obviously, you all live with this every single day. Medicinal chemists have a lot of parameters to optimize, particularly in the lead identification space, as you have many, many, many molecules to look at. And so we know that bioavailability is dependent on solubility. Great, we all get that. So then you can begin to think about how do you want to address this in the discovery space. And you could look on one end of a spectrum that is solubility good and solubility bad. The more the better. And so you can use high throughput screening techniques to just push that to the extreme, which of course would no doubt bring deleterious effects to other parameters that one is trying to optimize. On the other hand, you could go out and be extremely detailed with respect to characterizing solubility, permeability, thinking about how will all of this impact drug absorption. And that there's lots of great models for that. And as a pharmaceutical scientist, we use these tools all the time, PBPK type models, for physiological based PK models. There's plenty of commercial offerings out there. And you can get extremely detailed. You get very good predictions of what would happen given a set of physicochemical parameters, again, like permeability, solubility, estimating particle sizes, and so forth. That, of course, is another extreme, which is relatively slow, relatively labor intensive when you're trying to look at tens, hundreds, perhaps more molecules at one time. And so how do you find a sweet spot here that allows the discovery team, the medicinal chemist, whoever's working on that discovery team, to think about how can you optimize for solubility in a way that can keep up with the pace of all the other screening that's going on? And can you get those structures? Can you get the moieties built into the molecule that will lead to that solubility? If you do the too detailed route with extreme levels of modeling, you're probably going to be too slow, and you'll kind of get what you get baked into the molecule. And if you have a solubility issue at the end, well, then it's it's baked into your structural series. So something in this middle is probably the right thing to do. And we would propose that dose number is a very good metric to be in this Goldilocks zone of just enough detail, but not too much. Can I move this? Okay, I'm going to move this picture over. It's out of the way a little bit. Sorry, 1 second, Dennis. I'm just going to minimize that. Okay, great. All right, so before we get into dose number alone, we'll talk about, let's just revisit the potential absorption limits for a small molecule. And I would say that this was very well described by Dimano and Gordon Amadon back in the early 1990s. And so there was a series of papers that they published that go into the mathematics of what's on the screen here really well. Great series of papers, really fundamental, and basically, it's all the mathematics behind a drug particle going down an intestine, dissolving, drug exiting the system through permeation through the enterocytes, and then assessing what's left at the end of that event, right? What passes, and understanding what is it that will limit absorption. And they did a really nice job in this work, breaking it down into 3 limit cases of which one can think of, rather than doing the more detailed math at any given time. And so some of these I've just sort of simplified again for the sake of time today. Dose number we'll talk about more, but it is consider the dose number is dose divided by intestinal volume. So that will be a milligram per mil, or a concentration that one would achieve, divided by the solubility of the drug in that media, in the intestines. So you can think of it as a concentration divided by another concentration, which is the actual solubility. And so in the limit case, where a dose number is greater than 1, the intestinal milieu is saturated. You have a limit there that you can't dissolve all your drug. Another limit case is absorption number. And so absorption number is proportional to permeability and surface area. So that kind of makes sense that absorption number goes up as a function of how permeable and how much surface area you have to absorb a molecule. Now, surface area would be generally fixed between preclinical and clinical species, divided by the GI transit rate. So if we think about its limit case, again, an absorption number of less than 1 is a situation where absorption is actually slower than the path down the GI. So that would be limiting. Dissolution number is, you can think of it as, so it again is tied to equilibrium solubility of a compound. So it's proportional to equilibrium solubility divided by the volume of your drug particles. So the higher the solubility, the higher the dose number. The smaller number of particles or dose, the lower the dissolution number. So the higher the dissolution number as well. And then the volume would be the intestinal volume, which again would be fixed. And in this case, the limit again would be the dissolution number of less than or equal to 1. You're not going to be able to achieve full dissolution of your drug particles on the path down the GI path. So those are 3 potential limiting cases. Now they're all interrelated. Again, I would guide us all to read the original O and Amadon papers, and there's a really nice simplified version of the papers by an author named Brian Roars, chapter 7 of one of the AAPS book series on developability, where he tackles this in detail. And again, it's a bit of a simplified version, more like what's on the screen. And so it's an accelerated review if you'd like to read it. So the output of this math, and again, my understanding is this is the exact same math that is in our commercial PBPK models that you can purchase software licenses for. But this is, again, for this lecture, it's a simple way to walk through it. What you can think about in this whole drug absorption limiting cases are contour plots. So this is an example of a contour plot. We have it in our 2022 JMedChem paper to help describe it there as well. So we have 3 units here. We have absorption number, dissolution number on the x-axis, and dose number on the y-axis. And this is an example. The way we set this up is this would be a 200 milligram dose of a molecule with about 10 times 10 to the minus 6 centimeters squared per second with an in vitro permeability. That was the situation. So that comes out to an absorption number of about 3. The details of that calculation, again, I lead you to the references. And then you can see here are these contour plots. So what you see is the dark lines are that reflects a fraction absorbed. So the dark line is 0.9. So that would be 90% of your drug absorbed, given a set of parameters of dissolution number, dose number, and absorption number. And all the way out to a very low fraction absorbed of 0.1, which would be 10% fraction absorbed. And so when you inspect this sort of a plot, pharmaceutical scientists do this, whether they plot it out this way or not, or use commercial PBPK software. This is what they're thinking of when they're talking about how would you change a formulation. In our case, we're thinking about designing a molecule from the start. So as you see, we've talked about dose number. We talked about limiting cases of dose number. So you can see that once you've approached a dose number of about 10, you can see, if you follow this line, you can see you're in a place where you begin, theoretically, to lose fraction absorbed. Right? You're going from perhaps 60% absorbed, 70% absorbed, to very quickly to lower fractions that approach 10% absorbed at high dose numbers as you approach 100, let's say. On the dissolution side, you can see that while you could have a very low dose number, which again reflects plenty of solubility to dissolve your dose, you could have particles of sizes that are too big, and you could have limited dissolution here, and that's what's reflected again. While you have a low dose number, if you have slow dissolution, you will again struggle to get a large fraction absorbed. So those are the mathematical treatments around the limits of oral drug absorption. So what is it that makes then dose number? Let's just go back and focus on dose number, because that is the point of the JMed Chem paper in today's Flash Talk. Let's talk about dose number. Why is that particularly valuable to concentrate on? Well, here it is. It's calculation shown again, and contour plots left up. In reality, the dose number is, you can design for a higher solubility molecule. The permeability is screened by high throughput methods by many pharma researchers. So the permeability aspect is handled by other parts of the research operating plan, but dose number clearly is related to equilibrium solubility, which of course will impact dissolution rate in the future. And there is no particle size when you're talking about the lead identification space. You have a molecule. That's all later for development. That's all for folks like myself to worry about later on as we move towards the clinic. And generally speaking, so solubility is the right parameter to focus on if you want one simple metric to focus on as you profile your molecules. So in the paper, we talked a little bit more about, well, what is a reasonable dose number, right? So practically speaking, as med chemists, how might you apply this as you think about profiling your compounds? So we went through the literature. We found a couple of examples. Bayliss and co-authors from GSK, I believe it was, put out a paper back in, I think it was in the 2010s, and they had laid out a slightly different way of thinking about solubility, and they had a metric of dose divided by intestinal solubility less than 500 milliliters. So those are just it's a slightly different way to look at the data, and so we can recalculate that to a dose number of less than 2. And so dose number of less than 2 associated with favorable oral bioavailability. Sagano, an author from Pfizer, the 2010s as well, in published papers, and in those papers suggesting that dose numbers of less than 10, an aqueous buffer would be a marker of favorable oral bioavailability. Even went further in some of those articles, talked about if you're on the way to 40 dose number of 40, that would be a situation where there's going to be a lot of formulation innovation that's required. And if you're above 40, perhaps that molecule would be undevelopable. BI, Ingerheim had published within this, there was a decision tree published from, there was a consortium that was called Orfido, that was an academic industrial consortium that got together to talk about these sorts of questions. And in that paper, they referenced less than 10 using not just aqueous buffer, but biorelevant media such as so-called FASF, which is fasted state intestinal fluid. So again, another group suggesting that perhaps 10 is appropriate. In a separate article, Janssen had suggested less than 5 or 10 for aqueous buffer. And then at Merck, we tend to think of 10 as well as a reasonable marker for a dose number associated with oral bioavailability. Now, of course, there's 2 components to dose number. One is the solubility and one is the dose. And so certainly one can very early on just set a dose, 10 milligrams, 50 milligrams, 100 milligrams. And that helps the team think about solubility in the context of dose. So it's not just a matter of, you may know something about your program. Is it an extremely potent molecule? Do you expect the dose to be low? If that's the case, then you can also tolerate a proportionally lower solubility to an extent. So that's a great way to think about it on a team. It's not just more solubility is always needed or good. There comes to a point where you only need so much solubility, right? And dose number is a great way to put that in context. So as you see here on the screen, there's multiple criteria exist across the industry that have been published in the open literature, but generally speaking, lower dose number is agreed upon as better. And does that carry forward into what we know about commercialized actual products? And so we in the paper, we also went and we had looked at, there was a review article by Robert Williams in, I think it was 2018, maybe 2017. And what he had done is he had looked at all the launched amorphous solid dispersion products since I think it was 2007. And so amorphous solid dispersions, as probably many of you know, that's a formulation tool that your pharmaceutical sciences colleagues may apply to an insoluble molecule. So you use polymer technologies to freeze the drug into a amorphous state. Then upon release into the GI, you have a transiently higher solubility than you would from the crystalline state. And so it's a way to try to lower dose numbers, practically speaking, in the GI for a certain amount of time. And so we won't go into all the details here on the slide for the sake of time, but these were some of the products that were highlighted in that review article. And what we did as authors is we just simply went and found buffer solubilities, anything we could find in the open literature, looking at the marketed doses. But you can calculate those numbers. And so what you see in general is that consistent with the models of the contour plots of the general dose number theory, supportive of what was shown on the last slide about general dose numbers that are deemed advantageous for oral bioavailability. You can see here down this aisle that when dose numbers are greater than 10, that's what you're seeing in these products. As best we could tell from the open literature, it makes a lot of sense why these might be amorphous solid dispersions. So it does validate to a large extent that large dose numbers, right, 14 greater than 100, greater than 100, greater than 100, about 10, a molecule that has lower pH solubility but at higher pH, a large dose number, and so forth. You see generally large numbers on this list. So when you see large dose numbers, it does tend to lead to, let's say, enabled formulation efforts that are required to achieve the bioavailability that you want. So we thought this was a useful way to look down. You can take dose number into a LI or an LO program. How does it play out actually in the clinic? And we think that this table speaks to that. This is my final slide. I think T takes it after this. Is one of the questions that comes up then, we've talked about dose number, great, solubility in relationship to dose, very useful. How do you actually measure it? I mean, as far as I'm aware, I believe that most researchers will get some sort of a high throughput solubility measurement on their molecules in the lead ID or the lead optimization space. And the question's always remained, is that predictive or not? This is an example from our colleague who's a co-author on the paper, Pierre de Blaine, one of the co-authors with Tief and myself. And this is analysis that we had done. And so I'll orient you now. On the x-axis is whether the high throughput measurement for solubility was, this is out of an organic solvent into an aqueous buffer. Was it less than 50 micromolar? Was it between 50 and 100 micromolar? 100 to 150, and then greater than, was it less than 150? So it's binning from less soluble on this side to more soluble. And the actual colors here then are the actual manual solubilities that were made, manual solubility measurements that were made. And what you can see that brown is certainly the least soluble criteria value set. So let's just focus on that. Green being really high solubility. And you see this clear trend where brown is you see a lot of insoluble molecules when the high throughput solubility was measured at low value. And certainly when the high throughput solubility values are higher, we do see a lot more of the green and the light green. So generally speaking, in our experience, yes, the high throughput solubility is generally trends what you will see eventually when you have enough drug, when you want to begin more development facing activities, maybe crystallizing that drug, understanding the true equilibrium solubility and beginning pharmaceutical development on that molecule. But generally speaking, this is actually useful. We talk about this in the paper with a couple of paragraphs. There's certainly been great reviews over the last 20 years for how to do this sort of high throughput solubility measurement. The questions you'll ask yourself are, should you use an organic solvent such as DMSO or not? Are you going to check your measurements in buffer or buffer and biorelevant media? Are you going to check at low pH for weak bases and higher pHs? Do you do it at room temperature or at physiological temperatures? And then when will you check the solubility measurement? Will you check it at 4 hours? Will you check it at 24 hours? These all have some implications to what sort of numbers you will get out of the test. People will debate whether or not there will be amorphous artifacts, because almost always in high throughput solubility measurements, you're introducing the drug as an amorphous material. So you can have that debate amongst with you and your team as to whether or not you want to actually assess the crystalline state of that molecule and the measurement. And that really is a team-based decision. you could think of reasons why you would want to do that, and other reasons why you might want to just pass on that and take the high throughput value at face value. Sorry, the high throughput data at face value, and just proceed. So with that, I think we pivot now to Teeth.
Yeah, thank you, Pete. I think this is a good slide to transition, and I think just to re-emphasize the message here, right? So we should not, at least the way we think about it here, is we're not looking at this as absolute numbers. We are like thinking, can we categorize compounds? Because we're in discovery stage where we have multiple compounds to compare. And in this case, at least what the guidance or the general trend observe is that we know that 87% of the cases, the high throughput below 50 micromolar is telling us that's going to be probably a low solubility compound in the manual setting. And then you have the opposite, right, above the 150 micromolar. So one could use this data or these categories to kind of rank order the compounds, based on this correlation that we have observed. But as Pete mentioned, every team, every program, every organization might have different correlations, and they might need to set up different cutoffs or criterias. And in terms of how we design a compound that has better solubility, I think they have several reviews, a lot of efforts ongoing in the literature and described. We're not going to go in detail here. I think guidance is depending on the program, on the type of molecules that you have. But parameters is molecular weight of your molecule, number of hydrogen bond donors and acceptors, lipophilicity, the pKa of groups that you have there, and of course, do you have ionizable groups that they can generate salts or co-crystals? And I think those all impact your molecules, all impact your series, and that can help you to design a molecule that might have higher solubility, than the previous one. So I would say a lot of guidance is in the literature already, and I think as a general, we'll just say that there is multiple parameters that can help or guide a medicinal chemist to improve solubility. And I'm sure everyone has their own personal experiences in programs that they have worked on, which probably they could apply to every program they operate on. So what I want to, next slide, please. Yeah. So thinking about the discovery setting here, and after the introduction and the details provided by Pete, is we all start from a screenings campaign probably, and let's consider an HTS in this case where we have multiple compounds, multiple hits, so multiple choices, and we want to advance those compounds towards the hit to lead, eventually have some more focus at the lead optimization, and eventually candidate selection or different nomenclatures here, which means is we need to select the molecule that we want to push to development. So if we bring it to the solubility kind of mindset or guidance, again, this is probably not new to our publication. It has been already in the literature. And if we do one more click, so at the high throughput solubility, it's really very helpful because you're balancing the amount of compounds that you have to give you some kind of data. The type of high throughput solubility has been already described by Pete. There's several methods, and we can discuss about the pros and cons. But as I said, there are multiple, not only publications, but also vendors that they can provide some solution to your problems in providing a high throughput solubility. The question is, when do you trigger manual solubility? When do you start getting a form assessment of your desired molecule, which means, is it crystalline? Is it amorphous? Can I get the crystalline? Can I measure now kind of the solubility in the relevant media? And of course, that means that increases kind of efforts from the synthetic chemist or the medicinal chemist to provide that crystalline form to our colleagues in the pharmaceutical team. And of course, the choices here comes as late as possible because probably the number of molecules that you can go through this exercise might be limited depending on the size of your organization and priority of your program. But looking at this plot, it's all timelines, right? So we're all looking to accelerate the delivery of our compounds towards the clinic. And at the early stage, high throughput solubility, it's probably faster. We have too many choices. And as we advance the program, I would see a general guidance is we probably are deciding on leading compounds. That means we have more focus, and more focus means now we can dedicate a little bit more time in well understanding the compounds that we're deciding to profile. And this is where manual solubility and for assessment can come But remember that this is all about the number of resources you have and the timeline. So manual solubility probably is the best assessment to be done, but impossible to be done, with hundreds and hundreds of compounds without significant investment. Okay, so here we really wanted to highlight kind of an example, the same that we have in the publication, in how would we think about it at discovery stage? And as medicinal chemists, we're always thinking about designing, making, and testing, and of course, analyzing that data in this known cycle. So here, the example and the plot shown here are multiple series for a specific program where we have solubility in FASIF, in this case, on the y-axis, and we have cell-based potency on the x-axis. And this probably could be a regular plot where you're like, okay, I want to see, what is active and soluble in a way of speaking. And I want to pick a series that probably have less solubility risk or less solubility issues, because as I advance, I want to have a good starting point, right? So here we have 5 choices, 5 series, different colors. But in reality, these are individual parameters. I mean, your cell-based potency is not related to solubility, neither the opposite. So you could have very highly soluble compounds, but very inactive, or you can have very active compounds with low solubility. And we all know that at the end of the day, we need to balance these parameters to find the good fit molecule to move forward. So in next slide, so same plot here, what we wanted to reemphasize with this Flask Talk is that those number, it's a very, I would say, we can call it maybe simple parameter. It's a parameter that correctly directs to development stage. And if you think about the application of this parameter as an MPO, because those number includes the dose, which in that calculation includes the potency plus the solubility, now you have a parameter that correlates both parameters that we had in this plot on the left. So on the right here, we really use what we call a QSAR, where is let's assume that all the compounds that we have, it's a dose of 50 milligrams, and leverage the solubility that we have measured to give us kind of a value on the dose number. And as my colleague Pete mentioned, usually the general guidance within industry is the higher this dose number, higher is the risk of having a small molecule, having oral bioavailability risk and absorption risk. So what we are targeting, applying what we just learned in the previous slide, is we want compounds, or we want a starting point that is below 10, a way of thinking. Again, this is just general guidance. I think every organization, every team might have a different cutoff. But what I wanted to show you here is now we have a different plot, this box plot, where we have the 5 different series. And we can see how the series 4 and series 5 are really good starting points, because generally the average is below that 10. So if solubility is really the question here, and we're trying to mitigate the risk of oral absorption, I think one could decide to probably prioritize series 4 and 5 to advance to the next optimization cycle, instead of series 1, for example, where we can see that the average is very, very high, which probably put us in a more undesirable stage. Again, this exercise can be done with different doses and see if the same correlation applies. But I hope this graph here, simplifies the conclusion, instead of looking at individual parameters, such as on the left, where you have 2 parameters that are not correlated to each other and might just lead you to kind of, I would say, the wrong decisions, in a way of speaking. So let's keep in mind those numbers. It's a very simple parameter that we can apply very early on, as we discussed, and that parameter evolves. But we also wanted to highlight some limitations to keep in mind. I mean, we have been highlighting that passive solubility is probably the relevant one, but we all know that we might have compounds that might not be, the most representative ones getting the passive solubility, because their solubility changes, depending on the gastric conditions versus intestinal. And if there is an impact on the pH, because your molecule contains some kind of ionizable groups, then maybe one needs to consider getting solubility data in different biorelevant fluids or different pHs, and put that into perspective. The advantage is that those numbers, you could calculate those numbers based on the solubility you have, and those, depending on the fluids, and you can make an assessment. Is it those number going really high now that I have a pH that is lower, or is it the opposite? And hopefully that can help the team to make the best decision on those molecules. In the same lines, weak bases, and I think all of us here, we have worked with compounds that they are weak bases. We do know that the pH changes between gastric conditions and the intestine. So as you can imagine, those weak bases will have different solubilities. And I just mentioned, it's a matter of measuring solubility across different fluids and different pHs to get a good sense on the changes there. Another calculation or absence of calculation that was early on mentioned by my colleague is permeability. So those number does not include permeability. And in this case, what the team recommends always is, of course, get a sense of the permeability of your program and your series. And there are a lot of high throughput assays. There are even in silico methods right now. But definitely, if your series has low permeability, maybe those number needs to be taken in a consideration on a different assessment, because those number might not be reflective of the reality due to the low permeability. And finally, we know that food can have an impact not only on oral drug absorption, it can also have an impact on pharmacokinetics, and this is not different from those numbers. So it's not considered in the dose number calculation. So probably one needs to think about it more carefully when dealing with food effects.
I'll add one more. I don't think I hit this point particularly well on the slide with dose number and the contour plots. The other thing that I would say limitation, though it's infrequent, is when you have solubility that begins to get into the 20s and 10s micrograms per mil, that dissolution number can become a realistic limitation. And so the assumption of dose number is fast enough dissolution, i.e. dissolution number of greater than 1, so that it's not impacting your actual absorption. So you can just leverage the equilibrium solubility as the predictor. So as you get really insoluble, perhaps that's another limitation, is that now dissolution number may become limiting. So just another thing to consider.
Good one. Yeah. And finally, I mean, we're in last minutes here, the Flash Talk, but some take-home message. I mean, those number, it's a quantitative metric. It's really used to kind of get an assessment on oral absorption risk of small molecules. So I know there are different modalities being explored by different organizations. So I would say do not use our guidance to be applied to those modalities, or at least use it carefully in that sense. Again, it's a parameter that can be calculated as early as possible, leveraging in silico methods, leveraging high throughput data, manual, and that depends on every team. Just keep those assumptions in mind, because things can evolve from high throughput to manual. And the same is if your organization has a tool to predict human dose early on in your program, that could be already leveraged. Otherwise, our recommendation is assume your desired human dose and maybe use it to calculate different scenarios to give you kind of a sense of the dose number. And this way you can put into context solubility and as well as dose, instead of looking at the absolute individual parameter. We think this is a great parameter that can use between the discovery and development organizations. Our colleagues, and I learned a lot from my colleague Pete, where now we're using the same kind of language, and that can help us to kind of not only discuss about risk, but also how can we start thinking about potentially the risking or mitigating some of the challenges we might have for a molecule, instead of thinking, let's leave this problem later on for our development colleagues and they will solve it. I think as medicinal chemists, we understand the risk we're having and start interacting with our development colleagues. It can help us to just go a little bit faster as we hit those milestones. Pete, anything to add here?
No, I think you nailed it.
Good. Well, and of course, we cannot finish this. I mean, I think we would like to acknowledge all the co-authors in this paper. I know they're not speakers today, so hopefully we represented all of them. We thank them for their contributions. And I say, very happy, excited to be here, Denise. So
Yeah, very nice talk, both of you. Yeah, I think, yeah, dose number. I've read this paper and I was immediately excited. I think you both hit the nail in the head. It's like the earlier you start those conversations between discovery and development, I think what you said, Abdellatif, the faster you can move a project forward, and the risk that you go in the wrong direction becomes a little bit mitigated. Yeah, so fantastic talk. We already have a lot of questions here in the Q&A box. So let's start with that. And Abdellatif, you already kind of started talking about this a little bit when you mentioned the limitations. So here's a question from Chris John, and he's asking, dose number uses intestinal solubility as a key parameter for the calculation, and many drugs are basic amines, which will inherently have higher solubility at lower pHs, as you pointed out. And do you have or can you disclose any SGF dose number equation for those scenarios when the pH is 2 or in that ballpark? Is the dose number equation the same for those types of drugs that are more basic, is I guess the question.
If you want me to.
Yeah,
Okay.
please, Pete. I think,
Okay.
yes.
Yeah, sure. Okay, yeah, great question. You know, we don't address this in the paper directly, and I bet that each company thinks about this slightly differently. But the limit would obviously be if you do dissolve in the lower pH of the stomach. The question is whether or not you precipitate in the rest of the GI. And so I think that the idea of dissolve, those limit cases of do you dissolve all your drug or not, becomes, I think it remains the same. It generally would be intact. I think most people view an 8-ounce glass of water or 250 milliliters as a reasonable volume. And so one could think about that at pH 2 or 1.8 or whatever your gastric simulation would be. And then you could do studies where you try to address whether you can estimate whether the molecule would precipitate quickly or not. So I think you would generally remain intact. And if there's undissolved drug with a weak base, then you'd have to think about that perhaps in the context of what we just spoke about, if we just thought of the intestinal fluids. And so if only a little bit dissolves at low pH, well then predominantly you would want to consider this with respect to intestinal solubility. If it's 100% dissolved in the low pH, well then maybe that's when the precipitation questions, you want to evaluate them yourself. So those would be my thoughts on it.
Awesome. Yeah, I think that's super helpful. We have another question here from Anne Tran, and she's asking, do you recommend incorporating dose number alongside the VLE, which was another metric introduced by the Merck DMPK group, to help prioritizing chemical series in the discovery stage, and things that those parameters, dose number and V, complementary? Any thoughts on that?
Yeah, I think both are multi-parameters, that discovery chemists or medicinal chemists can consider, of course. And I think the more tools you have, right, the more data you can gather on your series, and you can assess depending on the risks or priorities of your program. So I would say yes, definitely they are complementary. One probably informs in a different aspects than the other one. And making decisions on which one to prioritize is really based on the program you're trying to solve and which questions you have and in mind that you're trying to. But I would, if I have the choice of having multiple ones, I would just include them probably in my calculations, and then help me to guide as problem changes, issues changes. So I know solubility, in my experience, you want to hide from it, but it comes back. So you want to face it early.
Yeah, that makes sense.
Yeah, and you may, if you're in the VLE space and you're thinking about it, maybe you don't have doses predicted yet, you could probably use the triaging case, right? If you know your potency, you will know a potency, some sort of an in vitro potency assay by the time you're listing these sorts of MPOs. So you could just 25 milligrams, 50 milligrams, 100 milligrams. I'm sure the team would understand some estimate as to what they think their dose would be at that time. So I would just peg it at some dose, and then that's how you calculate it, even if you don't know for sure.
Got it. Yeah, very helpful. Thank you. Another question here from Patty Meltson was, it was mentioned that dose number is used to predict oral absorption risk for small molecules. How well does this metric translate to beyond rule of 5 molecules, let's say macrocycles in the range of 750 to 1000 Dalton?
Pete, I think that one will be better suited.
Sure. I would expect not well, because I mean, I think if you were to go back to the absorption number, whatever your permeability is, I would assume your absorption number would become extremely unfavorable, and so you could have a very low dose number and a very high dissolution number, and you would predict very little absorption. So I guess so in that respect. So yeah, it gets more complicated because absorption, like Tief had spoken about, when absorption becomes extreme in these cases, it will perturb these parameters to the point that no longer is a good dose number just enough to get the absorption you need, right? There's a leap of faith that you have something, again, I'd say anywhere from 5 to 20 to 30 times 10 to the -6 in your typical in vitro permeability assays. This idea that the dose number being predictive generally holds. So once you go outside of that, I think you'd be at risk of just assuming dose number is the right thing to do. But looking at the dose number as predictive.
That makes sense. That makes sense. And do you see, Brian Marin is wondering, do you see a correlation between high throughput solubility and equilibrium solubility often, or is that depending on the series, I guess? So.
Yeah, I would say that we have seen that certain series are more predictive than others. And I think, Tief shared the plot. I'll go back to it, the one that showed our analysis of our own data sets, and it's on the screen now. I hope it's up for everyone. And that, yes, generally speaking, you see the general trends at the bottom, where, if our high throughput data sets suggested it was very low solubility, it was very rare that we came forward with no, this molecule is more soluble than we thought it would be. And perhaps that is potentially more valuable than the positive side, which is on the far right, which is if we predicted it was soluble. And that high throughput solubility number is prone to some error, because maybe it never crystallized in a 4 or 24-hour window. And then when you crystallize it, yes, the equilibrium, the true equilibrium solubility will be lower. So you would think that the far right would be more prone to error than the left, because if you're insoluble and amorphous, you're still going to be insoluble once you crystallize. But on the right, if you're amorphous and soluble, once you crystallize, the solubility could drop. But in our practical experience, it still generally serves. You see how the green is still, there's a big portion of green on that far right side. So is that an extremely scientific answer? I think it depends on the structures of your molecules. Certain molecules will be more prone to crystallize quickly, some that'll be prone to crystallize more slowly. And so you can think about that with your team and to figure out if your particular series are uniquely prone to errors and high throughput solubilities to predict the final equilibrium.
And just to quickly add, again, this is not about absolute numbers. This is about ranking compounds, right? So being in a bucket. Here we used 50 and 100, but a team, different team, different organization might use different cutoffs and still build some kind of comfort that they're ranking compounds in the good space or in an undesired space.
Yeah, makes total sense. Yeah, we're almost coming up on time. Maybe one more question. It's from Gagan. Is there any distant connection between dose number and predicted human dose?
Only in the sense that so dose number would correlates to fraction absorbed, and not, which correlates to bioavailability. So I suppose that if you had a extremely high dose number, and let's say, you might then say, well, I expect to get less absorbed than if your dose model was assuming 100% absorption, and your dose number is, I think the Sagano reference is a great one to read. He's saying, hey, if you're more than 40, you're going to really potentially be undevelopable. That's probably because your fraction absorbed will be, let's say it's 0.1, it's 10%. Well, that maybe would be a 10-fold change in your dose if you had predicted your dose on 100% absorbed, or 1.0 fraction absorbed. So in that respect, yes, but beyond that, I can't think of a connection between the two. Tief, can you think of a different one?
No, I think one includes the other parameter. So I think from an efficacy perspective, the dose is set right on your PK and your potency, and then you use that to calculate your dose number. Now, if your dose number is telling you, as Pete mentioned, it's like, hey, we need to get more compound in a different settings to be able to achieve those concentrations that you need to be efficacious. Then, of course, now you you know, your pill size or whatever, like your dose, clinical dose might change because you want to address those other issues. But I think, you know, it's kind of like a triangle correlating each other. So I don't think you do the dose number and now your dose magically changes. I think the, you know, the efficacy and the PK, it's usually the main drivers from a human dose perspective. Then of course, how do you make sure that you achieve those in the clinic? That might lead to some changes in your dose, just because you want to make sure that you're not losing any compound, or, you know, you just want to achieve that. So yeah, I wouldn't say directly one will inform the other one, but I think it might help to kind of triangulate in that perspective of thinking, okay, what is the end product in a way of speaking? But
yeah, sounds good. Yeah, thank you so much, Abdellatif and Pete, for this really, excuse me, this really great talk. We have actually many, many more questions, which I think speaks for how interesting the talk was, and that people really are very interested in that particular topic. Maybe we can get back to those later, to those questions. But yeah, I want to thank our speakers, Abdellatif and Pete, really for a phenomenal talk and sticking around actually over time to answer the audience questions. Really phenomenal job. I learned a lot, although I read the paper. So thank you so much.
Thanks for
Yeah,
the question, everyone. Yeah, thanks for the
yeah.
opportunity.
same here. Thank you so much.